Hi, my name is Annika, and I decided to do Jennifer Doudna for my biology role model. Some background about Jennifer: she was born February 19th, 1964, in Washington, D.C., but she spent most of her life on the island of Hawaii. She was always fascinated by nature, and especially Jim Watson's book about the double helix that her father had given to her. Some other sources of inspiration for her were her teachers and professors. Growing up, she faced a lot of adversity and was bullied, but this pushed her to do better overall. After she graduated high school, she attended Pomona College in Claremont, California. While she was there, she was accepted for a research position that really inspired her and got her interested in research and lab work. Then she transferred to Harvard and eventually UC Berkeley. When Jennifer's not doing research, she enjoys gardening, specifically growing the fruits that she had eaten as a child in Hawaii. And if she had to choose other than lab work, she would be a papaya farmer or an architect. She also enjoys reading books for fun, but she did note in an interview that it was difficult to balance work and personal life, so she had to put some of her hobbies on hold during the extensive portion of her research career. But when COVID hit, she found herself revisiting these hobbies. Why do I admire Jennifer? Well, I've always been passionate about medicine, and the Cas9 gene can alter these mutations that are responsible for the diseases. This could be life changing for patients experiencing debilitating diseases because it could ultimately be life changing and get rid of the diseases. Jennifer is deeply passionate about her career field, and I find that very admirable. She values diversity in science, she's humble, and she gave her work her all instead of shying away or quitting when encountering challenges or difficulties. Furthermore, her work opened a whole new world of discovery and research, which I find amazing. In terms of what field she's involved in, Jennifer Doudna is involved in the fields of chemistry, molecular and cellular biology, and genetics. The main question her research revolved around was if we could edit genes to modify genes in DNA or in organisms. Her most recent primary literature article was published this January 2024. It was about creating a molecule that effectively and efficiently delivered the Cas9 gene to a specific target. They did this by engineering enveloped delivery vehicles using gene editing machinery. As you can see on the image or the figure on the left, on the bottom are the target mutations that they were hoping for the molecule to target efficiently and effectively. And on the y-axis, it's the percentage of the envelope delivery vehicles that were able to target that specific mutation. And as you can see, based on the bar graphs, more than or about 75% of these envelope delivery vehicles were able to successfully reach these mutations, and only those mutations, which is amazing. And this article was called In Vivo Human T-Cell Engineering with Envelope Delivery Vehicles. What has Jennifer Doudna accomplished, and what's next for her? Jennifer Doudna pioneered gene editing research through the development of CRISPR, or the Cas9 gene. This discovery has caused a breakthrough in the scope of scientific capabilities, and the Cas9 gene can be used to alter disease-causing mutations in genes, which could be life-changing for many patients suffering from diseases. What's next for her? Oh, and by the way, she received a Nobel Prize for this discovery. What's next for Jennifer? Well, making CRISPR technology more efficient and safe. In the article that I had mentioned before, they talked about testing CRISPR on non-immune cells and checking the impact of EDV dose on T cells. Lastly, cures and treatments for human disease. I mean, Cas9 is revolutionary and could potentially save lives. Here are the references that I used throughout the video, and the images. The references for the images are all linked beneath each image I used throughout the video. Thank you so much for watching my video about my biology.